Professor Mohali. He will be talking to us about iron-depleted diet and how that enhances the lifespan of C. elegans.
Yeah, so first of all, I would like to thank the Academy for giving me this opportunity to talk about our research. As I stand between you and lunch, I must talk about diet, right? So I'm going to talk about the effect of diet on aging and lifespan. Yeah, so humans have a desire to live forever, right? Humans want to acquire immortality, and that is evident in several mythologies. For example, here you are seeing this white hair from the Chinese mythology, who is brewing the elixir of life, or a potion which can impart immortality. In Indian mythologies, obviously you would have heard about Amrit, and if you drink it, so you'll get immortality. But the fact is that humans have not been able to acquire immortality. The only certainty that is there in life is that everybody is going to get old, age, and die. Right? That sounds like a very sad start to my talk, but I'm sure things will get better from here on. So because we cannot acquire immortality, at least what we can try to do is, can we slow down the process of aging? Can we increase lifespan? So yes, we can. So that's a good news. There are actually several factors that can affect lifespan. For example, genetics is there. So that means we already know there are genetic variations that can lead to different lifespans. But genetics is something that is not in our hands, right? What genes we inherit from our parents is not in our control. But then there are things which are in our control which can modulate aging. All right. For example, whether you do physical exercise or not, what kind of food you take, these things can actually affect your aging, right? And what I'm going to talk about today is diet, role of diet in affecting lifespan, right? So the question that we are chasing is, can we identify diet components that enhance lifespan and eventually try to understand how they are doing so. Before I dive into this question, obviously you must understand that while we are trying to solve problems for humans, we can't actually do research on humans, right? For several reasons, and especially in our case, if I want to study lifespan of humans or how to modulate it, I will consume my lifespan in studying that, for just one person. For that reason, we use model organisms to understand the basic biological principles. And this is a list of some very commonly used model organisms. Obviously, a model organism means a well-characterized organism which has a lot of tools available for research. Some examples are, of course, different kind of bacteria such as E. coli, nematode or C. elegans in our case that I'm going to talk about. There is yeast, there are fruit flies, and so on. And of course, this list by no means is an exhaustive list. We already heard about zebrafish in the morning, and there are so many other model organisms. The model organism that we use for our studies is actually C. elegans or nematode. I'm going to just briefly describe what this is. So it's a free-living nematode. Free-living means it's not a parasite, right? So these are about 1 millimeter long worms, which have typically they are self-fertilizing hermaphrodites. That means they produce both gametes within one organism and produce their progeny. So the important point for us is that they have short lifespan of about 3 weeks, right? Because we are interested in studying lifespan, this helps us a lot because the lifespan is short. And they feed on bacteria, because bacteria is their diet. That also gives us an opportunity to modulate their diet and to study the effect on lifespan. The nice thing about biology is that a lot of basic principles are actually conserved. What happens in, let's say, C. elegans also happens in our body, right? So we can basically derive whatever information that we have and maybe apply to other organisms, including humans. Right, so how do we study the effect of diet on C. elegans lifespan, right? As I already told you, for C. elegans, bacteria is the diet, right? And so it feeds on bacteria. What we have to now do is, if we want to study effect of diet, we have to obviously change the diet. And how do we change the diet? Because it's bacteria, we can actually make mutations in bacteria. What we can do is we can knock out a single gene at a time, which will basically modulate the bacterial composition a little bit. We do that for all the genes, one gene at a time. Once you have these bacterial mutants, we feed C. elegans on them and measure their lifespan, right? So in that way, if any of the mutants changes lifespan, we can go back and look what mutation was there, try to understand what what kind of metabolic change might be there, and eventually find out what was there in the diet that was affecting lifespan. So that's the idea. And the nice thing is that we already have a single gene knockout library of E. coli, where all the non-essential genes are knocked out one at a time, which basically gives about 4,000 mutants of E. coli, right? So now we have these 4,000 E. coli mutants, which we can use, feed C. elegans on them, measure its lifespan, and see which of those are affecting lifespan. But it's not a straightforward job, because doing lifespan requires still about 3 to 4 weeks, and for each mutant we actually take more than 100 worms. So it's actually a very tedious job. So it's impractical to actually do lifespan on all the 4,000 E. coli mutants. What we instead need is, we need some proxy to lifespan, which can be used to basically find mutants which may affect lifespan quickly, and then you study whether they can affect lifespan. And that proxy in this case is basically lipid metabolism. Roop already talked about fatty acids in the morning. So basically these are long hydrocarbon chain with one acidic group at the end. We obviously have saturated fatty acids as well as unsaturated fatty acids. In this case, I'm talking about monounsaturated fatty acids. It's already shown that monounsaturated fatty acids can actually increase lifespan. Saturated fatty acids we normally obtain from animal sources, dairy products such as cheese or butter. Monounsaturated fatty acids we obtain from plant or vegetable oils. But inside our body, we can actually convert saturated fats into monounsaturated fats, and the enzyme that is involved in this process in C. elegans is called FAT7. So it was already shown the activity of FAT7, because FAT7 can modulate the level of monounsaturated fatty acid, is actually linked with lifespan. So Priyanka in my lab basically decided that I will basically look at the activity of FAT7, which can be done much faster than actually looking at the whole lifespan, on the whole 4,000 E. coli mutants, and whatever mutants we get which change FAT7 activity can then be used to study whether they affect lifespan or not. So that was the idea. But the question now is, how do you study activity of a gene? I'm going to explain this especially to non-biologists. So the expression of a gene, the expression basically means formation of RNA from DNA. The expression is actually driven by the promoter region, right? So the promoter region is a region which itself does not make RNA, but it actually tells when and how much RNA should be made, which is downstream of it. Because what really matters is the promoter and not the gene itself, the sequence of gene itself. What one could do is replace the gene sequence with something that you can visualize. In this case, it's a fluorescent protein, which is a green fluorescent protein, or GFP. So by using this strategy, you can actually visualize expression of any gene in live organisms. I'm giving you one example, or I'm showing you one example where this particular gene activity, which is ACDH1, and don't worry about the name, the point to remember here is that if you look at fluorescence levels, so these are worms which are showing you fluorescence, and these are fed on different bacterial diets. So you can see that on this diet they have highest activity of this gene, on these diets they have lowest, and somewhere in between in these diets. So the point is that you can reliably measure activity of a gene by creating such reporter strains. In our case, in this particular study, what we have done, we created a FAT7 reporter. FAT7 is, remember, the gene which is converting saturated fatty acid into monounsaturated fatty acids. This FAT7 reporter was used, and then we basically grew these animals on the whole 4,000 E. coli mutant library. This of course is much more fast and robust than measuring whole lifespan, because here we have to just see whether the fluorescence levels change or not. By doing so, we were looking for mutants of E. coli that affect C. elegans FAT7 expression levels, right? Once we find such E. coli mutants, obviously the next thing would be whether, because this is just a proxy, we actually are not sure whether whatever will change the expression levels of FAT7 will also change lifespan. So then we would like to see from that small list of E. coli mutants that are affecting FAT7 levels, will there be any effect on the lifespan? In lifespan, how we measure is basically you are looking at survival with time. Obviously, you start with all live organisms, so it's 100% survival. With time, some of them will start dying, and eventually you will see that the percent survival will come down. And by looking at the difference in the survival at somewhere in the middle, which is 50% of the animals are dead, you can actually see whether there is a difference in lifespan or not, right? Once we get this, we would obviously like to go back to the bacteria and see what kind of changes are there and see how diet is affecting lifespan. So Priyanka did this screen, and we found no E. coli mutant that enhance FAT7 expression levels, but we did find 26 E. coli mutants out of 4,000, which repress FAT7 levels, right. Just to give you an idea how that repression looked, this is wild type bacteria, this is that FAT7 reporter strain that we have. These are the GFP levels that you see, the fluorescence levels that you see, and on these 4 different mutants, these are just representative mutants. So here this is the gene, and the delta means this is the gene that is deleted, right. So these are the mutants where this particular gene is deleted. The names are not important here. The point is that we did find mutants which were suppressing the fluorescence levels compared to the wild type bacteria, and this is quantified here. Obviously the million rupees question is whether these 26 E. coli mutants that we identified, are they affecting lifespan or not, right. This is, we started with the assumption that this will act as a proxy to lifespan, but is it actually doing that? And very nicely we can see compared to control bacteria. So remember this black one is control bacteria. So these are C. elegans, their lifespan on the control bacteria. Compared to that, these mutant bacteria are actually having, or C. elegans is having an increased lifespan on these bacteria, right. So that actually shows that yes, we can use this as a proxy to lifespan, and we have found E. coli mutants which are increasing lifespan. So initially we tested a few, we were obviously very excited to see that they are increasing lifespan. Then Priyanka and Ravi actually screened all 26 E. coli mutant, did triplicates and measured the lifespan. So what you are seeing here is the delta T50, the change in lifespan. Obviously it is positive, that means the lifespan has increased, and as you can see on all of the diets we see that the lifespan was increased for C. elegans, and in fact on some of the diets you can see the lifespan is increased almost 50 to 60%, which if you think about humans, if somebody lives for 80 years, you are adding another 40 years to their life, which is huge. Obviously the question now is, okay, what is there in these diets which is leading to this increased lifespan, right. So first what we thought of doing is, we thought let's look at the changes in the gene expression in C. elegans which are fed on this diet, and see if we can get some idea what is happening to the physiology of the worms. So for that what we did, we took 4 different E. coli mutants and looked at the genes that were upregulated, which means their expression levels were high when they were fed on these diets. And what is very striking here is that a large number of genes is actually common, although these were mutations in different genes which were probably doing different things. A large number of genes is actually common, and only very small numbers are unique to each diet. So that shows that very likely all of these diets are doing the same thing or very similar things. And we looked at these genes and looked what their functions are, and it turns out that many of those are genes which are actually involved in mitochondrial function. So that suggested that maybe on these diets the animals have stress in mitochondria. So mitochondria are the powerhouse of cell, they produce energy, and if there is some stress in mitochondria, obviously the cells have to defend mitochondria, so they activate expression of some genes which can relieve that stress. HSP6 is one of the genes which is activated by mitochondrial stress, and just like FAT7 reporter that we had, one could create a reporter for HSP6 and use that as a marker for mitochondrial stress. That's already done. We used this reporter and looked whether the nematodes had mitochondrial stress when they were fed on these mutant diets, right. And as you can see, this is the HSP6 reporter which is reporting mitochondrial stress. This is the control diet, right. You barely see any expression of this because there is no mitochondrial stress, but on all of these 26 diets you see that there is increased expression of this reporter of mitochondrial stress. So there is indeed mitochondrial stress on these diets. But now the question is, okay, what is leading to all these phenotypes? Why is there mitochondrial stress, and how is the lifespan has increased? So we looked at literature. We also considered the phenotype that we had taken together. It turns out that the E. coli mutant that we had, when obviously we looked at previous studies, what kind of changes were there in these diets, it turns out that they have low iron levels, these 26 E. coli mutants that we have. So then we started testing the idea, could all of this be happening because of low iron levels, right? If it is because of low iron levels, then if we supplement iron on these mutant diets, we should see reversal of all the phenotypes that we had so far. Obviously the 3 phenotypes that we had, one was FAT7 levels. On these mutant diets compared to the control bacteria, there are low fat levels, FAT7 levels, but the moment we supplement iron, you see that now there are those fat level low levels of the FAT7 are rescued. Likewise, when we use the mitochondrial stress reporter HSP6, again in absence of any iron supplementation, on these diets there was mitochondrial stress compared to the to the wild type bacteria, but the moment we supplement iron, all those differences disappear, right. More importantly, when we look at lifespan, remember this is in absence of any iron supplementation. What you can just focus on instead of worrying about this, just look at the delta T50, which just tells you that there is 30% increase in lifespan on these diets. The moment we supplement iron, again just focus on this plot, which is derived from this plot, you barely see any change in lifespan in these diets. So that means iron is able to actually rescue the phenotype that we were seeing, and if it's really iron limitation that is leading to all these phenotypes, we should be able to create the same phenotypes in wild type bacteria if we are able to limit iron in wild type bacteria. So for that we used an iron chelator. So this iron chelator will basically limit iron levels even in wild type bacteria. So now we are looking at wild type bacteria in absence of iron chelator and in presence of iron chelator. You can see that the FAT7 levels indeed go down. This is the mitochondrial stress reporter, it goes up, so there is mitochondrial stress, and most importantly there is an increase in lifespan when we limit iron even in wild type bacteria. So all of this shows that indeed limiting iron levels in the diet are increasing lifespan of C. elegans. Finally, we ask which of the host genetic pathways are involved in this mechanism, and so I'm basically, this is a busy slide, but don't worry, I'm going to explain what is this. So each one of these is actually a mutant of a genetic pathway that leads to increased lifespan in C. elegans under different conditions, right. Here what we are seeing, we are checking whether a given pathway is actually required for increased lifespan. You can think of this as if, let's say in whatever hotel you are staying, you had to come to IISc and there are 3 roads, right. If I block a particular road, I want to know whether you came through that road or some other road. So if you don't reach IISc, I know you were trying to come through that road which is already blocked, but if you still came to IISc, that means you came through some other road, not the road I am thinking about. So this is the same thing, we are just blocking this pathway by having these mutants. If these pathways were required, you will not see any change in lifespan, but as you can see, compared to the wild type bacteria, the mutant bacteria increase lifespan of these mutants and these mutants. Don't worry what these pathways are for now, but in these 2 specific cases you see there is absolutely no change. That means these were actually required. So what are these 2 pathways? This is a pathway that is involved in response to starvation. So whenever there is starvation, this pathway gets activated and try to defend against that starvation. This pathway is involved in energy sensing, so sensing energy levels, right. Both of these are actually related to diet, so that's very exciting to see, because we were all talking about diet and iron levels. So to conclude, what we have seen is that iron depleted diet can enhance C. elegans lifespan, and this happens through pathways that are involved in sensing starvation as well as energy levels. What we would like to do next is because so far we have seen only iron is regulating all these phenotypes, we want to know whether there are any other metabolomic changes in these E. coli mutants which can also affect lifespan. That is one thing, and of course we would also like to see if the same thing happen in other models, right. So with that, I would like to thank my lab. Priyanka is the one who has done most of this work, and Ravi helped her, and I would also like to thank funding sources, and thank you for your patience. Yeah.
Talk is now open.
Thank you for beautiful talk. Now I have a question that you're talking about FAT7 reduction. So can it be tested by a blood test or something?
So we did measure, so in this case the reporter was a protein reporter, in the sense we were actually monitoring the protein levels, so the enzyme levels are going down, but we have also measured the metabolites, which is metabolite is oleic acid and stearic acid, and those levels also have changed.
What does your study, is it complementary to Cynthia Kenyon's kind of work on the extension of lifespan of nematodes and also possibly of humans by restricting the amount of diet? I couldn't quite see, because you didn't mention any of her earlier work. I was just wondering, what does that have to do with this?
Right. So, to give a context to all the audience, so Cynthia Kenyon's group has actually looked at first genetic pathways such as insulin signaling. Downregulation of insulin signaling leads to increased lifespan. Other studies have also shown that if you restrict the amount of food that is given, or calorie restriction, that can also lead to increased lifespan. So there are different genetic pathways involved in all of these. In our case, we are actually initially we didn't want to study the genetics per se, we wanted to see if diet affect that. So that's something that is different from what Cynthia Cannon has done. And secondly, I would also not call it as calorie restriction, because iron probably won't really provide any calorie as such. So it's also different from calorie restriction in the sense that we are limiting iron.
It certainly seems very complementary, right? I mean, you're finding definite pathways that are extending lifespan,
Right.
and these are metabolically controlled,
Right. Yeah, so I mean, at genetic level, as I said, there are multiple pathways involved. So there are specific pathways which are required for iron-mediated increase in lifespan, which are different from insulin signaling, for instance. The HLS30 pathway is actually common between dietary restriction and iron starvation.
Pardon my ignorance, but I was just observing that if you look at the multivitamins that is given to people at 55 plus, 60 plus, 65 plus, and 70 plus, one finds that as years go by, the content of iron is decreased, and at 65 plus onwards it is minimal. Does it have any scientific basis based on extending this study, or is it just random people have observed this way and then giving it?
Yeah, so the reduction in iron level with age could be, a decline in ability to absorb iron, and that's why vitamins are supplemented so that iron uptake can be increased.
No, what I meant is at 65 plus, the iron uptake is minimal.
Yes.
I mean, the extra iron given in the supplement is minimal compared to, let's say, 55 plus.
Right.
Any correlation with this extension of lifespan?
So you are saying, could the reduction in iron uptake with age affect age?
Yeah,
or aging or lifespan?
Yes.
Well, I can't really comment on that because I don't know if we know about
this is what is observed.
This is
just by the dose.
So see, iron levels are like double-edged sword, in the sense that if iron levels are too low, obviously that's going to be harmful. At the same time, if iron levels are too high, that's also harmful because it can lead to ferroptosis or oxidative stress through Fenton reaction, which is also bad. So there has to be a balance. In our case, I don't know if C. elegans already get more iron than what it needs. So yeah, I don't know that. Yeah.
Hello, sir. Nice. Sir, have you compared with other bacteria which are present in the microgut of?
Other bacteria means other than these 26 mutants that
No, other species, not only E. coli, because normal flora of the elegant, the what you find and how you selected E. coli is one model.
So in lab, E. coli is the most commonly used diet, right? Of course, there are other bacteria other than E. coli which exist in the gut of C. elegans in natural environment. Until now, so far we have not actually used other bacteria, right? We are only focusing on E. coli. The reason is because there are mutants available for each gene. We can control that.
Yeah, that's fine, sir. You have used availability of the mutant.
Right.
My question is there already gut is there, the elegant? How they are going to be interactions?
Okay,
so
in lab we grow C. elegans only on E. coli. So in their gut there is only E. coli. There is no other bacteria that is present.
Okay, fine, sir.
Yeah.